Atrimed lab. Morning, everybody. Hope I'm audible to you all. We will start in another 3 minutes at exactly at 11:06 because there are many people joining. So we will have enough numbers. Yes, let's start. At the outset, let me welcome on behalf of Atrimed Pharmaceuticals as the MD and CEO of this company. I'm so happy that there are several people, a few hundred people who have gathered here on our, this WebEx meet, as well as many people watching uh, on YouTube and live on Facebook. This is actually something revolutionary what we are trying to do, and I'm very happy that you're all part of it. I request you to be part of it, to make it successful by participating in it, by talking about it, by giving your best to make good data, scientific data for us. This is the first time that an industry and academia joining program has been started by in our field, which is very common in other fields. This industry academia program, what we have started is only as a trial run now, which will be big in coming years. That basically means that the budgets are going to go up, the number of colleges or participants are going to go up, and the data we create is going to go up. It's very important for us in this world to survive, to create data on efficacy of our products, as well as create data to prove that our medicine, that's Ayurvedic medicine, is less toxic or non-toxic. But then we have to create data for that, and for creating data, all of us have to come together. So this data needs to be analyzed. We have to confirm either the efficacy or non-toxicity and publish in peer-reviewed journals. Once we publish these in peer-reviewed journals, we ourselves get confidence, plus we can shut the mouth of those apologists. Now, first question comes to everybody's mind is why house surgeons? Why didn't you extend it to everybody? In fact, this is open for everybody. Here, I don't mind a house surgeon, MD scholar, lecturer, or even a physician who is practicing outside to be a part of this, study. But the focal or fulcrum of this particular study will be a house surgeon or an intern for a very specific reason. House surgeons are enthusiastic, and they are budding doctors who are going to come and start practicing in next 1 year or 6 months, and they have a lot of time in coordinating things with various others, whereas MD scholars, for example, will be very busy with their own thesis, their own work, and they may not be able to dedicate their time for clinical or preliminary work like ours. By the way, we today's program of training you will focus on mostly of clinical trials, whereas Dr. Lata, who is going to speak, will also talk about preclinical studies. We are open for any type of study. It can be a preclinical study, it could be in a literature search, it can be a clinical study, it can be an animal study based on available facility. So since house surgeons are fulcrum, they can join hands with the lecturers, and they can join hands with PG scholars who can advise them. But then the maximum clinical time available is for house surgeons. That's why we have chosen house surgeons, and we are open for even changing that rule. But let's stick to that for the time being. Now, many of you may be thinking that when we get into trial, like you need enough numbers to prove that efficacy or non-toxicity. So many of if you choose a disease, there's a possibility that you don't get enough numbers within the period that we are talking. It's around 3 to 6 months or maybe 8 months. So we can do multicentric studies. That is, if we see that there is a very well written protocol which can be applied in 3 to 4 colleges, we can do a multicentric study, and we will be willing to support that. Also, many of you may have doubt that this is only 1 lakh rupee that people are going to give. Is it going to be enough for me? And for the study, I mean, we are flexible about it. We can club, and instead of doing 4 studies, we may be doing 3 studies or 2 studies, or we may increase our budget if we find that some of you have written fabulous protocol, which is very significant. So don't be disheartened. Go ahead, go write a protocol which is close to your heart and which you feel that it's going to definitely win this competition, and you are going to get funded. Now, apart from you doing clinical research, what you're going to get is you're going to get access to Atrimed. I will tell you, Atrimed has got 4 to 5 labs. We have I think the only Ayurvedic company which has a computer-aided drug design, that is computational chemistry lab, is with Atrimed. Here, you can virtually screen any plant against any disease. In fact, you can screen multiple plants against one disease target, and we can narrow down the possible leads that we are going to have for clinical, our study. You will have access to our molecular biology lab. You will have access to our genetics lab, confocal facility, microbiology lab, cell culture lab. So when I say this, this may be corollary to your, clinical, your research work that you are planning. As you go around, when you plan your research work, there'll be a lot of inputs for you from our scientists. We have cell biologists, we have microbiologists, we have synthetic chemists, we have plant chemists, we have Ayurvedic doctors, we have genetics, we have all of them who will be helping you to improve your protocol and make it such a work that you will be proud about publishing this in peer-reviewed journals which are of international repute, as well as who have got good value. So if you want suggestions, you can, while writing the protocol, you can reach us out. We will be willing to help you, unbiased. We have three prominent doctors as our main jury. We'll be unbiased about who has to get selected, etc. We will not definitely not uh, interfere. Other than those three, we have Dr. Suresh from Kerala, Triveni Nursing Home. We have Dr. Shenoy from Mysore, who's extremely good at protocol writing and especially about statistics. If you have to choose the right number of people as a participants, then he can really help you, and he can help you prior to selection as well. And we have Dr. Pratibha Nadig. Pratibha Nadig is DM in clinical pharmacology, very ardent supporter of Ayurveda. She is the HOD at Dayanand Sagar Medical College, Bangalore. She will be also talking today about various aspects of protocol planning and research studies. We have with us Dr. Lata Damle. Lata Damle is graduate in Ayurveda, MD, and she has also done some fellowship in Japan. After that, she has done her postgraduate degree, that is MS in drug development. She is versatile with Ayurveda as well as preclinical studies, as well as clinical trials. So she will also be helpful for you. So it's going to be a good journey. We will do some phenomenal work, and it's not just chemistry alone. We will be also giving you. We want to do some, basic work also, and Dr. Lata will be giving you some of the ideas. How can we work on, basic science like basic principles of Ayurveda as well? We can do that. We can. There is no limit. You can do toxicity studies, field trials, literature search. We do not want to limit to anything. Any good study coming out of this, we'll be very proud of. And as I said, we can continue the study later as well. Don't be worried about time limit. Even if you finish your time, of house urgency, you can still be part of the study. Will we till we finish this study? So we are flexible, and we truly want to be neutral, and we truly want you to triumph and have a wonderful time generating data for our Ayurveda. Thank you very much. With those words, I welcome you all, and I request Dr. Lata to take over. Thank you.
Thank you, Dr. Damle. Hope I'm audible. Am I audible to everyone? Just let me know, one of you. Okay, so after that insightful talk by Dr. Damle, I'm sure you all are very energized and very excited to put your brain into good exercise, right? So here I would like to give you some insight about the way we can go ahead doing research and some ideas to kickstart. So, and I also would like to urge that this particular platform, take this as an opportunity or a stepping stone to transform your thinking in the direction of vigorous and innovative thinking, okay, in scientific works, in the future also. So I want to share my slides with you now. Everyone can see my slides. Somebody can unmute and just tell me. Okay. Okay, good. So first of all, what's this research all about? Research must be the most cliched word you all would have heard, right? Everything has research, researched cloth, researched textile, or any researched juice. But what is this actually research? Research is basically a mindset actually. So I would say I always like to give these two examples when I talk about research. The first one, this is for the reason why we need tools to verify our observation. Of course, observation can be very beautiful, but is it good to put it to acid test? Okay, so this one simple example proves that it is important. You must have seen these illusion actually. It's an illusion. Even if I say it's an illusion, at the end of the day, even if you believe that it is not true, in the mind you want to believe that both these lines are of same length, right? So all we need actually is a measuring tool or the scale to tell you that this is an illusion and both of those lines are of the same length. So that's what scientific tools do actually. Next one, you must have seen this. This was actually done, I think for a long time till Galileo did this experiment. Many would think that balls of heavy weights, the one mass, the object of heavy weight would reach the ground faster. But when it was shown that both would reach, of course, in vacuum without being affected by air pressure or air, the breeze, they would both reach the ground at the same time. But for this, actually, all you had to do was carry out that experiment. Just hold two objects of different masses and just drop yourself and see. You can see which one falls faster. So this is how what you need, how the thinking should go. That's what is basically research is all about. It's not just holding test tubes or working in lab with the lab coat. It's basically the thinking or the mindset. So the science itself, I would say, it's not the knowledge. It is basically the best tools that you have, which actually can be put to whatever that you have observed or objective, the subjective observations to quantify and to objectify it. So these should be used as tools to test all our hypotheses or whatever we think. And once you start doing that, in every aspect or where any hypothesis you come across, you will start analyzing in this critical fashion. So it's basically a methodology of thinking and working. And very importantly, the three qualities that it can have is demonstrability. That means you can show that it is, yes, when I drop the balls, yes, they fall in the same speed. Then reproducibility, which is very important. Whether the other person who repeats your experiment or who follows your hypothesis also has the same result. Then comes a very important is fallibility. In any critical thinking or in any kind of hypothesis that you put and enumerate, you should think whether I can be fallible. Yes, fallibility is inherent quality of everyone, including the science. That's why it sharpens its tools continuously. Many a times we feel that science is stupid, that today it said something, tomorrow it says something. No, it's actually sharpening its tools and making the observation, understanding it critically and giving you better result every day. Because the bias can easily enter in our observation. When we see how you saw the Muller-Lyer illusion, however we dismiss, inherent bias can always come in. That's when we say we need to design the protocol. That's what is all this exercise about. How you can do the research protocol design devoid of bias. How we can avoid the bias, possible bias. I'm sure more details about this. That's why the statistics is all about actually, which will be given by Dr. Shana and Dr. Pratibha will talk more on this. I just want to tell few types of very important bias that can come is information bias. When you start collecting data which is inaccurate. Or it could be selection bias. You're not randomized the selection at all. Or you have selected wrong category of patients. where you are not supposed to give any medicine. For example, I did see one of the study where they had included pregnant women in the study. So what happened, in fact, pregnant women should not be included in the first setting because ethically it is not allowed. Such kind of selection, lack of randomization or wrong selection can make your design poor. Third one is very important, confirmation bias. Confirmation bias is, I think, very common. When we like something, we'll try to gather evidence in a way that it supports our view. For example, if you like coffee, you will get all the data or evidence that says that coffee is very good for aging. Okay, that's one thing. So this is what we tend to do when we design the protocols. So these are the bias which we need to avoid. So how can we avoid? Of course, there's no first thing and foremost thing would be we should be self-aware and honest about what we think and how we can design, where we can go wrong. We can plan that well ahead of study. And well ahead, planning well ahead always gets you good outcome. That's very important. So take more time in planning so that you don't falter at later stage. So have a good clarity on objectives, clarity on the outcomes, how well you can blind and remove the biases, also address those earlier three sources of bias that I said. As I told, this will be well explained during later sessions by Dr. Shana and Dr. Pratibha. So in this particular research protocol competition, we are concentrating on 4 ideas. Of course, you can come out with more ideas. These are just pointers. Research is a vast topic and sky is the limit. You can always come out with better ideas, and that's all about, it's all about research actually. So we have, to make it things a little more easier, we have suggested 4 kinds of arms to this research protocol, which will either you can take up on Atrimed products. Okay, Atrimed products also, in fact, we have made it a little easier, have given you some suggestions of the products, but you can take up any product and you can work on that. Or you can think of some basic science principles like Siddhantas and come out with some design a study on that. Or you could go in for an Ekamulika Prayoga or a single drug medication. Or apart from all this, you can come out with any novel ideas you think can be useful. And in all these 4 groups, you can choose any in silico, in vitro, in vivo or clinical trials, depending on the feasibility. You must also, because while designing, you should also think about the feasibility of the portion also, not just selection. Okay. So you can plan accordingly. So in the first segment where we have Atrimed products, we have suggested 7 products. Of course, these will be recorded so that you can go back to this and look later as well. Either you can think about a combination drug of Viscovaz with the Zeptico. These are basically for wound healing and secondary ulcers. Then could be Spasix, which can actually work as a single drug remedy for asthmatic attacks. So you can choose either it is an acute case or the chronic case. Of course, you also have to plan the duration of the study because I'm sure in housemanship you have the constraint of time. So think about and what best parameters you can use. For example, whether you will use pulmonary function test before and after. It could be just 2 months study or 3 months, whether there is any improvement in the pulmonary function. So another could be Aclear, which is again, the reason why we have also thought about this is these can be quickly done. For example, acne doesn't require many number of days to get cured. Similarly, you can have kinase, which is for pain management. You can have pain scores and manage design accordingly. or it could be an upper respiratory infection or acute rhinitis, flu-like syndrome, for where you can use Viridol, and it could be just 5 to 7 days of treatment. And or you could also think of anti-dandruff remedy, Scurfol. Try that. Or you can think of any type of psoriasis where you can use Atrisor ointment and Atrisor capsule. More details of its dose and other things, you can definitely check the product details, or you can come back to us as well. Some suggestions in basic science. These are, as I am saying again, these are just pointers. You can come out with your own ideas. You can take more help from your mentors, tutors, because we have best of the brains out there. So you can get many informations and you can come out, design a very good study, I'm sure. So just to put some pointers, I have given some examples here. You can think of role of Viruddha Ahara. But what I would suggest is, since the time period is short, I would suggest to be specific about what you want to expect, or what the primary objective or outcome should be. So role of Viruddha Ahara in immune response, or in the inflammatory response of, whether it has any impact. You can study on that. Or is there any relation between the way we respond to inflammation in our prakritis? Or is there any relation between rasa guna virya vipaka that correlates to class of phytochemicals? For example, if you have madhura gana, or these kind of virya guna virya vipaka, do they all come in terpenoids? Do they all come in, say, if you have some bitters, tikta rasa dravyas, do they all come in alkaloids? So these kind of class of phytochemicals you can assess. Of course, not just literature review, you can test it using HPLC, HPTLC, and such other instrumentation. Similarly, you can also, you must have seen, we have 20 fresh plants. The Ayurveda suggests them to be used as fresh drug. So you can really see, by using them in fresh form or in the different extracts, is there any difference in the phytoconstituents actually? Or is there any single drug versus gana? When there is if you see, there are groups of single drugs. For example, Vayastapana gana or Sanyajana gana. So if you take single drugs, each of them, do they have any similarities in the LCMS peaks, or do they have any differences? What is it? That you can study. Similarly, can Samaprakriti person have a differential immune response? Does he have a better immune system? Of course, this needs good amount of blinding with many people assessing the Prakriti, so that we don't need to, we don't have contradictory assessment. Similarly, you can also think whether the absorption of drugs on Pushya Nakshatra will be better compared to any other days. I'm just giving Pushya Nakshatra as one day. There could be many. If you see Kaumara Vritya or any other Samhitas, you will get, this has to be given on this particular time. It is well mentioned. So you can study those and come out with ideas. Upavasa is another, Langhana is another very important concept that you can study on. You can see whether there is any inflammatory or allergic response with the blood parameters like ESR, CRP, IgE or IgG responses. Also, you can think of Shodhana effect or effect of Vasti on gut microbiome. We can do the microbiome of the feces, genomic analysis, and find out that. So these were just the pointers. As I said, you can come out with much better ideas. Similarly, next set of studies could be Ekamolika Prayoga, that is single drug usage. So this, you can have ideas from Agre Dravyasthana, where single drug is said for, most of the time, for example, Rohitaka is said for liver ailments, but many of the medicines that we use in actually liver complaints does not contain Rohitaka. Of course, there are some, I'm not saying there are no, but it's hardly, we have not seen this kind of single drug being used in that particular where they have said agria. So you can really see whether different dose forms of the same drug can have differential results, or same drug in different types of those, jwara. When they say jwara, is it all kinds of jwara, or are there any typical pattern to it? You can study those things, or you can get some idea from, as I said, ganas also, like vayastapana, kandugna dravyas. Are they specifically, why have they been put in one particular gana? Are they all have similar effects, or they have any different patterns of different kind of disease with similar symptoms? That you can study. Similarly, you might have seen one haritaki is being told either, either as a powder, it has different usefulness. When it is boiled or steamed, it has different efficacy. Actually, does it have any differences of phytochemistry also when it changes? We can check that. Whether the efficacy difference also occurs, of course. Really, the one is used in Atisara, one is used in Gulma. So you can check whether there is a difference in efficacy as well. Not to forget Anupana, because with the different Anupanas, the effect seem to be different in as per text. That also you can put to test to see that is possible. Well, doing this, planning this is one thing. Designing to have a proper outcome is also another thing. Last, very important, is novel ideas. Of course, I don't need to put any guidelines or I don't need to give you any pointers, because then that never becomes a novel, right? But still, it is, I do have some novel ideas. If somebody comes out with that, I'll be very happy. So just give you some pointers which you can think of. Can you make self-dissolving bandage for non-healing ulcers? Say you put the bandage. We do have, in fact, biodegradable bandages available now, made of silk actually. So can we incorporate medicine in that and make a self-dissolving bandage for non-healing ulcers? Can we see whether we do use Kshara Sutra, right? Can we develop something, a liquid that solidifies as it goes, traverses through the Bhagandhara fissure tract, and probably is more easier to apply? Is it possible to have some intranasal medication to treat CNS disorders, or even some systemic disorders through intranasal? Because we have whole host of medications suggested in Nasya. And of course, some very novel thinking where we don't have a good answer at this point of time. Can you really stop that hair follicle destruction? Because that happens not just part of aging, it's more to do with the hormones. Can we have something in the last phase where it destroys? Can we inhibit that particular enzyme so that hair follicle destruction can be stopped? Or can you have any other methods of tissue culture, or aeroponics or hydroponics and such other systems to have sustainable delivery of active compounds? Why I say this is because for every medicine we need to use a lot of plant sources, right? So we need to make at one point of time these medical compounds, the active compounds, sustainably available. So can we think of such? Of course, I'm sure you will come out with more ideas. So at the end, I want to tell that we are here to help you. So I just gave you some pointers. As I told earlier, we can do all these works, either take it as in silico work or take it as in vitro. You can plan your studies accordingly. Pilot studies, blind, randomized control studies, or anything. But at the end of the day, I want to assure you that we are here to help you, and we want you to excel. And take this as an opportunity to have a innovative thinking in future also, so that we get much more fruitful ideas, and well, we can harness the beauty of Ayurveda. Of course, it's a competition. I'm sure we have made it competition only to make you more active, But as I said, work with the vision to continue forward the scientific thinking. Thank you very much. Wish you all very good luck. If you have any questions, you can ask me now, or you can write to us later, however the time permits. Prithvi, do we have? Hello? Yeah, I see a question here. Research is to be done. Final year students can definitely join. But one thing is, I'm not sure if interns are allowed to prescribe drug. I don't think so. You will have to have the supervision of your teachers and professors in your college. Pop in your questions in the chat box if you have any questions. If no question is there, maybe we can move on to next speaker. I see one question. Will date of submission of protocol be extended? The current deadline is January 15th. I think we have enough time. You still have some more than a month to prepare. Because if you extend still more, if you don't submit at least by Jan 15th, I don't think your 3 months or at least 4 months of study can be finished. Yeah, everyone can see there's an answer there. Stage 1 submission starts from 15th December and the last date for submission is 15 January 23rd. You can choose from any of those 4 topics that I just mentioned. It will take some time for the next speaker to start his session. So probably till then you can think of questions if you have any. And we'll be available here to answer the questions. You can either open your, you can unmute yourself. Or let me know if you want to switch on the video and ask. Yes, this is going to be recorded session. You can watch it later also. I will check with the people here how they can send you the links to view the recorded version. The link will be sent to you. There's a question here where we have to submit the protocol. So we will send you the link for that. Oh, I'm so sorry. Okay. Uh, this is a problem. It's very difficult to look at an inanimate object and talk to people. I don't like this. It's better we have at least some faces visible. Um, I was talking about Dr. Dharmaraj's question. Yeah, it's a very good question. You are asking about modifications in medicine. That means you are asking whether we can have different dose forms like injection or IV fluids. Definitely, you can think of dose forms. But only thing is, you will not be able to ethically allow to test it anywhere. You will have to have lot of preclinical works before taking it forward. But you can definitely plan the study, the basic studies for that in this particular protocol. And for those who wanted to know where the submission has to be made, it is at www.atrimed.com under research. I think most of the people are being admitted. I don't think anybody is waiting. You can also study PK parameters. For example, when I said single drug or combinational treatment, you can see whether when you combine the drug, what is the bioavailability of one particular marker compound. You can test in patients. You can see whether they are in the serum, the bioavailability of one particular active marker is being supported or being increased with the addition of other drugs in the kashayam or group of medicines. You can try those studies. It can be done in vitro or in vivo also, actually. In the rat model studies, you can do. Or in healthy volunteers also, if it is a very common drug that we use. Using just ginger alone or tricotue, the combination, does it make it potentiate the benefit? That you can see. Whether any marker that you think is not very desirable, like a toxic compound, whether that goes down if you add the other drugs. You can study those kind of works. Similarly, these can be, you can plan in silico studies. I don't know how many of you have done an in silico study where we study the interaction of drug with the protein of interest or the target protein and see its binding reactions, whether it really binds to that particular. For example, you can think of a disease and the well-established targets for that. For example, if you say alpha glucosidase enzyme in diabetes, if you want to study some anti-diabetic drugs which can have this particular activity, that we can try in silico first and get a clue, and then you can proceed for in vitro studies. But when you think of any study, as I told, first plan the study well. Discuss with your mentors, discuss with your tutors. You can also reach out to us for any help regarding designing of the study or whether even ideating process, and then assessing the feasibility, whether we can really take it forward, such studies. Looks like there are no questions, or probably thinking process has started, I guess. Think where you can make difference. And when you choose this study, I would suggest use those critical thinking when you, because recently I saw one study where they said, the sitting on the ground and eating is good because when you bend, it presses on the pancreas. It might press on the pancreas, but doesn't it press on the kidneys also then? Right? So I think you need to be critical when you claim. I'm not saying that this is not good, but is it really that good? Is it really, is another thing affects? You need to think critically. If there are any questions, do post in. I think you guys might be already bored seeing me alone. Probably the other speaker will join us soon. Till then, I keep my video and audio switched off. Okay, the moment now we have some question, ma'am. The aspect of dosage of Anupana isn't mentioned in our Samhitas very precisely. Is there any scope on doing research regarding that dosage of Anupana? If my understanding goes right, we do have different Anupanas. First, I think you can see whether those Anupana, the relevance of those Anupanas. Now, whether I use 1 spoon of honey or half a spoon of honey, it is discretional call. Most of the things, I think, sometimes we have to think logical also. When the Anupana is mentioned in Ayurveda, definitely our ancestors wouldn't ask you to take glass of honey, right? So it is that much amount that is needed to probably mix that particular drug. I think we should take that logical and first assess the basic relevance of that Anupana. Then maybe you can definitely modify the drug, whether you need only just 1 drop or 2 drops or 5 drops. You can definitely, right? I hope that answers your question, Gayatri. So as I said earlier, I'm sure you are getting bored seeing me all over the screen. I think I will switch my video and audio off for a while now before the next speaker speaks in.
हाँ सर आहे. हाँ हाँ ಅನ್ಮ್ಯೂಟ್ ಮಾಡಿ ಸರ್. ಮಾಡ್ತಾ ಇದ್ದೀನಿ ಸರ್ ಶೇರ್ ಮಾಡಿ. हाँ सर. ఇప్పుడు Hello, sir. Good morning, everyone. I'm Dr. Fareen, first year PG from Panchakama Department, JMC Mysore. Dr. Shenoy sir will join soon to this presentation. Today, I'm here to present about the research planning. So, the content of the research planning are introduction, literary review, hypothesis, aims and objectives, methodology, ethical consideration, and statistical analysis. In this presentation, we'll learn the process of the carrying out the research. Okay, first we have to understand that research is nothing but it is a process of transforming knowledge into information and information into the wisdom. So in this whole presentation, we'll learn how to carry out how to carry out how to carry out a research project and what are the steps involved here to make it to go through a what are the steps involved here. So I like to tell you the basics, the steps involved. First, we have to do is define. First, we have to take a topic which we find any difficulty around it. That we call it as a research problem. After the research problem, we have to do the literary research, and after that, we have to make an hypothesis based on the literary review. And after the literary review we have to do collect the information and study design have to make based on the type of research is done. Then after that, okay, sorry. Sorry for the inconvenience. I like to start with the introduction of the research planning. Research is a process of collecting, analyzing, and interpreting the information to answer the questions. Research is a structured inquiry that utilizes acceptable scientific methodology to solve any problem in a any problem. According to Clifford Woody, research comprises of defining and redefining a problem, formulating hypotheses or a subject or suggested solutions, collecting, organizing, and evaluating the data, making deduction and reaching to a conclusion, careful testing and the conclusion to determine whether they fit in the formulating hypothesis or not. In the whole process, from we start with a problem which we have difficulty to understand. So in the whole process we think about that problem, and in our order we start inquiring about that, then we analyzes the data what we have gained, and finally we come to a conclusion whether the question we arise whether the question we arise whether it is true or false. So the whole process of research consists of all those things. The steps involved in the research process. The first is formulating the research problem. Second is extensive literature review, and third is the developing the objectives. Fourth is preparing the research design, including sample design, collection of the data, analysis of the data, generalization and interpretation of the data. Next is the preparation of the report or presentation of result, formal writing, and conclusion the things by writing a design. Okay. So okay, the first step of research planning is formulation of a problem. Formulation of a research problem include, it is the first and most crucial step in the research process. The research source include the formulation of problem should be we should follow two things. First thing, it should have four P's, that is whether it is people oriented, problem oriented, program oriented, or phenomenon oriented. Then while writing a problem, we have to check some checklists, like whether the question is feasible, whether it is very easily available. Next is whether the problem what we have, whether the research what we are performing is interesting, whether it is new, ethical, and repetitive. By keeping all this in mind, we have to form a research problem. The next thing, another format we can follow is PICO format. That is, we have to check the population. The whole research formula, the research question should include population, intervention, comparison of the group, intervention being compared, and the outcome of the interest. Okay. The research process is a similar to undertaking a journey. For a research journey, important decision have to make. But first, what we have to do is while starting our research journey, what you want to find. The main question arises: what we have to find. Okay, so after formulating a problem, the next thing is the literature review. The literature review is an integral part of entire research process and makes the valuable consideration to every operational step. It's for what are the importance of literary research is. First, we have to gather all the information related to the inquiry with what we like to conduct. And the main function of literary research review are bring the clarity and focus to your research problem, to improve your methodology, broaden your knowledge, and contextualize all your findings. So it is very important while starting our research, we have to have a thorough knowledge about what the problem is, what are the present consequences, and what are the things told and what are the things not explained yet. And so we get a clear cut idea about the thing which we have, which we have to do in future. So the next thing is the sources of literary review. Actually they are the sources of literary review. So there are mainly three sources. The first one is the primary source, second one is the secondary source, and third is the tertiary source. So we have to understand what it is. So the first thing is the primary source. It provides direct or first-hand evidence. These are documents that provide full description of the original research. So these are nothing but the history which is taken by the investigator, or anything which is published in a peer review article, it is first-hand. That is with there is no any alteration in in that data. It is directly connected by the investigator. Such kind of data can be considered as a primary resource. And the secondary resource for literary review are these are the interpretation and the evaluation are data derived from the primary sources. So here, what we these are the interpretation. Like we gather some information about a patient. Then, based on that history, we like to conclude that maybe this causative factor is responsible for that disease. So that we'll keep in that we will keep that in mind, and we'll do further researches on that. So we have to understand the prior so secondary sources are nothing but these are the interpretation from the secondary sources. So, for examples like all the research articles all the research article and other conceptual articles, these are nothing but the secondary sources. So, last, what is the tertiary literature? This consists of a distillation and collection of all the primary and secondary sources. So the example like books which consist of both the primary and the secondary. The first thing what we have got from the actual person or from aptas, and the second thing the interpretation of the author. Both will be in the tertiary literature. So next is the concept computerized database like Ayurvedic research database. So we can search in all different kind of indexed articles, PubMed, Cooper, and gather information. It also come under the tertiary literature, or it depends on which kind of article it is. If it is clinical research, then it can be a primary resource, or it is any kind of conceptual article, then it can be a secondary resource. Even many general can be considered as considered under tertiary literature. So these are the three resources for literary review. Next is after first thing in the research planning and research process, the first thing is to formulate a problem. For formulating a problem, we should go with five things. It should be interesting and specialized, and it should be any ethics. We have to look over all those things. After that, after formulating a problem, when we get an inquiry, the next thing is doing the literary review. For literary review, we have three things, that is primary resource, secondary resource, and the tertiary resource. The third thing is after having a complete knowledge about a thing, and again we have a problem. The next thing is hypothesis. After doing literary review, we'll get an idea that at what point we have to conduct the research. That point is nothing but the hypothesis. So the whole process of research will be conducted based on this hypothesis, which is hypothesis is nothing but it is considered as a principal instrument in a research, as it is the first instrument in the research. Based on that, the further proceeding will be there. It is main function is to suggest new experiments and observation. What is the main aim of the hypothesis is it suggests an experiment and observation. The definition of hypothesis is a hypothesis is a contextual statement or the relation between two or more variables. Okay. So hypothesis is nothing but it is a statement which gives us relation between two or more variables. Here, two or more variables are nothing but the reason one is cause, another one is the effect. So we have to find the relationship between a cause and effect. So hypothesis does that. Hypothesis is a formal statement that presents the expected relation between an independent and dependent variable. So it is nothing but a statement from which will conduct our research, which define the both things, that the problem and the probable solution. Okay, the characteristics of hypothesis. So how should be the hypothesis? Hypothesis should be hypothesis are nothing but assumptions. So before starting a research, we have some assumptions, and at the end of the research, we'll get the result of that assumption, whether as a, what we have assumed is correct or false. Okay, the hypothesis should be clear and precise. Next thing, hypothesis should be capable of being tested. Hypothesis should be limited in scope and must be specific. It should be not very uh, hypothesis should state relationship between two variables. Hypothesis should be consistent with most known facts, amenable or testing within a reasonable time. Here the statement what we have to prove what we have to prove in the whole research process, it should be clear and precise, and even it should be capable of testing. Like the next thing is what are the main I what a hypothesis will do. Okay, what are the types of hypothesis? There are many types of hypothesis like alternative hypothesis, a simple hypothesis, empirical hypothesis, statistical hypothesis, complex hypothesis. So one by one, we'll learn about the different type of hypothesis. Okay, the first one is the simple hypothesis. What it is? It is a simple hypothesis is a statement made to reflect the relation between exactly two variable, one independent, another one is dependent. Okay, let me give an example. Here, smoking is prominent cause of lung cancer. The dependent variable here is lung cancer, and independent is dependent, and the independent variable is smoking. So here smoking may cause lung cancer or it may not. So this kind of hypothesis are considered as simple hypothesis. Okay. Next is the complex hypothesis. Here, in simple hypothesis, there were only two variables. One is dependent, another is independent. So in a complex kind of hypothesis, it is contrast to a simple hypothesis. A complex hypothesis implies the relationship between multiple independent variable. Example, for an instance, individuals who eat more fruit tend to have higher immunity, less cholesterol, and high metabolism. The independent variable is eating more fruit, while the dependent variable are higher immunity, lesser cholesterol, and high metabolism. So we can, with this hypothesis, we can conclude that if a person eats healthy, then he will have good immunity, and lesser cholesterol, and high metabolism. So here there are only there will be multiple variables. Next is empirical hypothesis. What is empirical hypothesis? It also referred as the working hypothesis, or empirical hypothesis claims a theory validation via experiment and observation. Say the hypothesis is women who take iron tablet face a lesser risk of anemia than who take vitamin B12. So how we can why it is called as a working hypothesis? Because we'll continuously check whether the woman taking iron tablets will have lesser risk of anemia by doing various kind of lab investigation, and compare with the other things like vitamin B12 taken. So here the question is, a woman will take both the tablets, iron tablets and vitamin B12 tablets, but through the lab investigation will come to a conclusion that which is one is better. So this is the comparative kind of research design which comes under the empirical hypothesis. Next is the statistical hypothesis. The point of statistical hypothesis is to test an already existing hypothesis by studying a population sample. Hypothesis like 44% of the Indian population belong to the age group of 22 or 27. Evidence to prove the disapprove of particular statement. Here in the statistical kind of hypothesis, there will be in the statement, there will be some ratios or some measurement. So it will be more numerical kind. So a research problem which is having some particular ratios or a definite measurement that will come under the statistical hypothesis. So the most important kind of hypothesis, which is very relevant to research, medical researches, and all kind of researches are these: the null and alternate hypothesis. Here, these are the two assumptions. First assumption is in first assumption, like alternative assumption, we try to prove the thing. In the null hypothesis, we try to reject the thing. So one will be positive statement, another will be negative statement. Here, alternative hypothesis is a positive statement, and null hypothesis is a negative statement. Example, the null hypothesis is a claim of no significant difference between two variables. And the alternative hypothesis is a claim. Here, it is termed as H naught, is a fast or opposite to the null hypothesis. Here, what will happen is, in a hypothesis, there will be two statements, two variables. One is dependent, another is independent. Like let me give example. Like Panchakika Niruhabasti will help, our hypothesis is study the efficacy of Panchakika Niruhabasti in the management of psoriasis. So, first, now this is the research problem. For this, alternate hypothesis will be the Panchakika Nirohabasti will help in the management of psoriasis. And the null hypothesis will be there will be no effect of the Panchakika Nirohabasti in the management of psoriasis. So the positive statement is alternative hypothesis, and the negative statement is null hypothesis. So we can see the difference here. In the null hypothesis, it is denoted as H naught, and alternative hypothesis, it is denoted as H. A statement about a population parameter. We test a likelihood of the statement being true in order to decide whether to accept or reject the alternate hypothesis. Just one thing we have to keep in mind that the both are contrary statement, and at the end we have to test the hypothesis and prove whether it is right or wrong, whether it is false or true. The null hypothesis assumes there is no difference between two groups. Example, let me give a good example. Null hypothesis is H naught. Here, light color has no effect on plant growth, and the alternate hypothesis will be light will have some effect on the plant growth. So you can see this both the statements are contrary to each other, and we have at the end of the research we have to prove whether the alternate hypothesis is true or the null hypothesis is true. Okay. After this is the fourth step, that is objectives of the research process. The first thing was formulation of a problem, which we have to define. The next thing is doing literary review over that. The third thing was forming an hypothesis, that is forming an assumption about our study. After forming assumption, the next thing is forming the objectives. Here, objectives are nothing but these are the blueprint of our research design, that what are the steps we'll follow in the upcoming research. Example, okay, the objectives are the objectives are the specific action or activities to answer the research questions. So these are the specific action or the activities which are taken by the investigator to solve a research question. Objectives indicate what we are trying to get from the study or the expected results or the outcome of the study. So, by just looking over the objectives of an article or a research, will come to any investigator, any person will come to know that what is the motto of this research and what will be achieved by the end of this research, and what way we have to conduct the result. Conducted research. Research objectives should be clear, achievable, and verifiable, as they direct assist in answering the research question or a problem. So, the research objectives should have three things. That it should be clear. That what are the findings we will get in this research, and achievable. Not like it should be too vast or too time taking. So it should be achievable in a limited period of time, as we'll get some limited period to conduct our research. So our objective should be achievable and verifiable. Next is the objective may be specified in the form of action. So while writing an objectives, it should be in a verb form, that it should be in action form. The words like determine, to establish, to assess, to identify, to compare, to analyze, and to evaluate. These kind of action words should be used while forming an object, forming an while writing an objectives. We should use action verb. Next is the characteristic of research objectives. So the like how the research problem was having PICO format or smart PICO format, finer format. In the same way, objective should follow these things. If your object is having object is having smart format, like first thing it should be specific. The aim of research should be smart, like S indicates specificity, M is measurable, A is attainable, R is realistic, and T is time bound. So our objective should be specific, it should be measurable, it should be attainable, it should be realistic, and it should be time bound. Okay. Okay. I think you will get a after getting an idea about the objectives. Again, in the objectives, there are two types. First one is the general objectives, another is the specific objective. What is the general objective? These are the broad goals. So objectives are nothing but the goals of the research or the steps involved in the research process. These are the broad goals to achieved and state what the researcher expect to achieve by the study in general terms. Okay, general objectives are nothing but our problem. The problem is nothing but comes under the general objective. What is the main goal of our research? And what are the specific objectives? These are the sub steps. Like these are the short term and narrow in focus. They address various aspects of the research problem, they should specify what the researcher will do in the study and where and for what purpose. They are divided in three phases like explorative, descriptive, and explanatory phases. So, while writing the objectives, there should be 2 points. One is general objective, another is specific objective. General objective is nothing but our research problem is our general objective. As a specific objective, let me give an example. If we are comparing, if our research problem is a comparative kind, and we are comparing the result of two dravyas, in that specific objective will be like to check the efficacy of the first drug, and second objective will be like to check the efficacy of the second drug, and the third objective will be like to check the relationship, to check the efficacy, which is better than, which is the better drug compare, which is the better drug, the first one or the second one, or to check the relationship between both of them. This will be our sub objectives or specific objectives, because we are in that okay, importance of objectives. What is the importance? First thing is it will focus the study to narrow it down to the essentials. Sorry. Avoid the collection of unnecessary data, organize the study in clearly defined part of phases, facilitate the development of research methodology, orient the collection, analysis, interpretation, and utilization of data. So just by putting some objectives, we'll come to know all of this thing. First thing, we'll focus over the study. Next thing is avoiding the collection of unnecessary data. When we have fixed some point, like when we fix some target, we'll only try to focus on that target. In the same way, the objectives will help in focusing, and it will also help in collecting the unnecessary data. The next thing is the research design. Okay, research design is very important aspect of research planning. Here in the research design, what we'll do is in what way we will conduct the research, how many samples will be collected, what will be the methodology, and which kind. This all will be based on the research design. So it is a detailed outline of how a research process takes place is called research design. Research design is also defined as it's an architectural blueprint of the research project or a conceptual structure within which research is conducted. In the research design, when we let me give an example. When we start to construct a building, first we'll make a blueprint. At first, we have to make the foundation. After that, we have to construct the first floor and the second floor. In the same way, research design is nothing but it is a blueprint. Please. Okay. Now we'll study about the different research designs. So what are the different blueprints available in research are? First thing is it may be observational or interventional. Second, it can be descriptive or analytical, cross-sectional or longitudinal, cohort study or case control study, control or non-control study, preclinical or clinical trials. As you can see in those pictures, and we'll get an idea that what kind of research, what kind of different kind of research designs are. First one is descriptive. Just we in the descriptive kind, we will do no intervention. We just look over the people, over a population, and conclude our results. Like it is just an observational kind. Next is experimental. So in the experimental, we do many experiments while conducting a research. Next is diagnostic and explanatory. Okay, let's understand what is a descriptive kind of study design. In the descriptive study, various type of surveys and fact-finding inquiries are included under the descriptive research study. So, in the descriptive study, various surveys, okay, any research which include any survey or the data collection is mainly based on the observation. And the second thing, there is no intervention by the investigator, then it is a descriptive study. Okay, there are three ways a researcher can go about doing a descriptive research project. First one is observation, defined as a method of viewing and recording the participant they are. So, in the observational kind of study, but the first thing, the research design will depend on what kind of research. It will mainly depend on your research problem. Based on your research problem, will decide will decide the research design. Example, our research problem is our research problem is just finding the number of people, the number of smoker will end up into lung cancer. Then it is just an observational study, and there is no and there is no intervention. Then we can consider it as a descriptive study. So, in the descriptive, observation defined as a method of viewing and recording participant they are. First one is case study. Again, there's a subtype defined as in-depth study of an individual or a group. Survey defined as a brief interview or a discussion with an individual about a specific individual topic. Okay. Next is the cross-sectional type of study. Here, what will happen? There is a population, and in the cross-sectional kind of study, the first thing is in this study, only one observation is collected at a time by a researcher. The process of this study is it is mainly the cross-sectional study will have two groups. First, we'll select a population with some similar qualities, or we'll select a population of an area. Then again, the population is divided into two subpart. Like one population will have disease, another population will not have any disease. So after when we do review, we'll get to know this population there is some risk factor. So in this two population will study whether the risk factor have any effect or not, or will give some intervention and check on both the group, the group which is having disease and another group which is not having disease. Okay, advantages of cross-sectional studies. It is cheap, rapid, and easy. We can use on large sample of population. It assess health status and health problem, and indicate priorities for healthcare planning. Assess customer satisfaction for healthcare. The next thing is provide the baseline data for further studies if the problem is not studied before. Next thing, cross-sectional studies is the most convenient first step in the investigation of the cause of the outbreak or epidemic. So all the survey study which we conduct are nothing but the cross-sectional studies by students. Like we will form some questionnaires, and only we go to the people and ask only once, or we collect data only once a time, and further we'll analyze the data. So cross-sectional studies are nothing but taking the information only one at a time. What are the disadvantages of cross-sectional study? The cross-sectional study or the survey study or case series studies, the sequence of event cannot be ascertained, not practical in studying rare disease. Case reports are susceptible to bias. Case report cannot be used to make treatment decisions. As we don't follow up here in the cross-sectional kind of study, we don't do follow-ups. Here we take data only one at a time, so that there might be bias about the data. So these are the few disadvantages of the cross-sectional study. Next is case report and the case series. In the unit of the study, okay. In the case report, there will be on a single. We'll do study on a single person. If our research is including or it is only on a single person, then it is a case report kind of study design. And the limitations are based on the experience of a single person. The first clues in the identification of the disease or adverse effect of exposure, description of of a polytrauma patient. Okay. So by doing only a single a research on a single person, we cannot come to a definite conclusion, as it will definite conclusion, as there is much population is needed to study the same thing on a different people. So next is case series. What are the case series kind of study design? It is a unit of here the unit of study are the group of the person with a similar disease. In the case series, we'll conduct the research on a group of people which is having the similar disease. But in case reports kind of study, we do research on only on a single person. And what are the uses here? The formulation of criteria for formulation of formulation of criteria for diagnosis, formulation of indication for the treatment. Okay, when we study. When we study a group of people having same disease, then we can formulate a criteria. We can formulate a criteria, and next thing is we can formulate the indication for the treatment, identification of the prognostic factor, and determining the survival rate. Just by studying a group of people, we can get we can get a good quantity of information. With that, we can come to a conclusion whether the survival rate of the person in the disease is how much is the survival rate of the disease in the person. Next is analytical studies. Analytical studies are nothing but it is an type of research. The researcher has to use information which is already available and analyze to make a critical analysis. Example, you can see here in okay, what are the examples of analytical kind of studies are case control studies, cohort studies, and randomized controlled clinical trials. So mainly what we do in analytical studies are we check the relationship between two variables. Example with a factor like the risk factor. We know this disease will uh, have some. We will know that eating. We will know this risk factor will lead to a disease. But we check the same thing in a two different population. One with the disease, another one is a healthy person, and we'll analyze that by when the person get exposed to that risk factor, at what when he will develop a disease, and we'll check whether a healthy person have gone through this kind of risk factor or not. So in the analytical study, we'll just check the relationship between the two variables. Okay. Okay, next another type of study design is qualitative and quantitative. Okay. The quantitative is the focus on a specific concept, whereas the qualitative focus on the entire phenomena. Okay. The main thing we have to understand, but the difference between qualitative and quantitative is if the first thing is the quantities are nothing but it will be objective oriented or number oriented, whereas the subjective criteria will be depend on the data which cannot be counted. So it is more the quantitative data are more objective kind, and the qualitative data are the subjective kind, where quantitative data will focus on a specific concept and qualitative focus on an entire phenomena. Okay, next, in the quantitative, we'll only focus on the ratio of liver lipid profile, and in qualitative, we'll understand the behavior of the person. What are the symptoms? What are the here we will focus on more about the thought process of an individual. Next, we begin with the preconceived thought about how the concepts are related. Okay. Next, use structure, procedure, and formal instrument to collect the data. In the quantitative type, as the data is very important, that too data which can be counted. You here use a structure, procedure, and formal instrument. Next, the collected information under the condition of control. Here, the minimal control will be there. Here, quantity will be more objective, it will be more subjective. And in the quantitative group will be focused on small group, and the qualitative will focus on a large group. More time in the quantitative data will take more time in processing of a data, whereas in qualitative kind, more time is selection of subject and the study. Next is the preclinical and the clinical studies. As a preclinical are nothing but animal studies, and clinical studies are nothing but a study involving humans. So the preclinical and preclinical study is to test a drug, a procedure, or an other medical treatment. Okay. As animal. Okay, in the preclinical study, what we do is we use animal for to check the efficacy of any drug, to check any adverse effect, to check to formation of the main thing is to develop a new drug. While developing a new drug, before checking its efficacy on a human and to check its safety, we conduct the preclinical studies. The aim of preclinical study is to collect data in support of the safety of the new treatment, the new intervention, or the drug is screened through the various stages in the suitable model of animals. A clinical study is nothing but a clinical study is a trial or research study done in human volunteers to answer specific health questions. Clinical study is also an interventional type of research which is designed to answer specific questions about biomedical or behavioral intervention or including new treatments. Okay, you can see the chart of randomized control trials. Here, randomized control studies include first, okay, you can just look over the different type of study designs. After learning about the study design, the next thing is collection of the samples. Samples are nothing but it's the representative subject of a population, which will represent. These are nothing but the representative of a population. Here, the samples can be collected in various forms. The first thing is non-probability design. Second is the probability design. In the probability, we'll check the probability design are nothing but um, these are nothing but the random sampling. Here will have an idea of the chances of the involvement of that person or the individual in that study design. So probability collection. Here, it is done usually in a rare disease where a researcher will where a researcher will get the individual by referral of their referral on the previous person. A person with a rare disease will refer a researcher another person. So this is a snowball kind of sampling. Another thing is quota. Here the researcher will divide the population into small groups and choose accordingly. And next is based on convenience. Based on convenience is nothing but the selective kind of sampling. When a disease is rare and the population ratio is very less, then based on convenience, the researcher will select the samples. And in the probability design, it is of more organized and it is considered as most important. So the the probability design consists of simple, random, systematic, certified. So now, now Shanoy sir will continue the presentation.
Okay. So thank you one and all, because sorry for the delay, because I had some meeting with my boss. So that's why I asked Farin to continue with the presentation. Thank you, Farin, for giving a wonderful presentation. So we'll discuss this thing. So welcome to the the presentation. Today we have to discuss 4 points. One is writing methodology. What are the steps to write methodology? The second one is ethical considerations, how to consider the ethical things. The third one is IEC, that is Institutional Ethical Committee. And the last but not the least is hypothesis development. So we will talk about these things. So I think Farin has covered many of the things. So I will summarize the things. Okay. The first and foremost thing for you is that how to write a title for the synopsis. These things are very, very important for you. So how to write synopsis title. So title should have 3 things. Title should have 3 things. One, title should have a problem. Title should have a design. Title should have a solution. So when you talk about a title should have 3 things. One is title should have a design. Title should have a problem. Title should have a solution. Now, you may ask me a question that what is meant by a design. So we will talk about the design. So you have to go to one of the important area. You have to choose that area, and based on the area, you will decide the design. So we are having many designs. So clinical area is one of the design. You can say I am doing work on clinical, so it is a clinical design. So we are having many designs. When I talk about clinical, we are having many designs. You can compare about 2 drugs, and it is called as comparative design. You can compare between a standard drug and a trial drug. You can compare between a standard drug and trial drug, and it is called as a controlled design because you are controlling and comparing with the trial drug. You are controlling and comparing with trial drug. We call it as a trial design. We call it as a comparative control. Comparative control. So when I talk about design, you have to mention the name of the design. There are many designs are there. So we are having sampling techniques. That is, you have told all those things. No. Okay. So you have to talk about design. Then how to identify the problem? So first of all, you have to identify the problem. Now question comes, sir, from where to start? How to start? What to start? When to start? All these questions, you should have these questions in your mind. When you talk about something, you wanted to talk about COVID. These questions on COVID. One, what is COVID? Then how it spreads? When, why, where, and what? So if you try to answer these questions, you will get to know that, okay, I am getting some good answers for some questions and I am not getting some good answers for some questions. If those areas are there, those areas are called as gap areas. You are getting some gap for that question. You are getting some gap for that question. I will give you an example. Mr. X is a handsome guy. Mr. X is a handsome guy. So that means you will get to know what is meant by handsome. Whether his height is very good, his weight is very good, or his personality is very good, or his dressing sense is very good. So you will try to list out why he is handsome. You got my point. When you get these things, then you will have a standard thing. Okay. Oh, to say handsome means these are the qualities I require. Out of these qualities, this person is having this much of quality. So you are comparing a standard and you are observing the characteristic in a person. So compare between these two. When you compare between these two, you will get some gap. The gap is nothing but your intervention. I will come to a disease. So say RNTCP. For tuberculosis, the modern people are going for an RNTCP program. The name of the program is RNTCP. So I will list out what are the advantages of RNTCP. I will list out, okay, RNTCP should be given for 6 months. RNTCP, there are 5 drugs are there. Okay, RNTCP given for 6 months. There are 5 drugs are there. Weekly thrice drug should be given. So you will list out all these things. Now you will get a question. Why I should give it for 6 months? Can I give it for 3 months? You got an opportunity. See, why there are 5 drugs? Can I do it with 4 drugs? You got an opportunity. Can I do it with 3 drugs? Can I do it with 2 drugs? Can I do it with 1 drug? See, you are getting. So you have to list out the advantages of the problem. That is called, we call in a scientific terminology, we call it as SWOT analysis. SWOT analysis means strength, weakness, opportunity, and threat. So whenever you find a problem, don't talk about the weakness. You have to talk only about strength. Suppose if I say this person is a cruel person, what you will get? You will get only the threats. Okay, I should not talk with him. I should not touch with him. I should not quarrel with him because he's wicked. He is a cruel person. If I say that person is very good person, you can talk. So that's why whenever you get a problem, try to discuss the problem in terms of strength. Try to discuss those problems in terms of strength. Then you talk about when you get the strength. Then when you go on discussing the strength, you will get the opportunity. So strength is there, opportunity is there. Try to do the gap analysis between strength and opportunity. You will get the intervention. So that's why write a title. Title should be always should have a design. Design I have given here. These are all the designs are here. So design, so it should have a design, it should have a problem. So how to identify problem? I have told, take any problem X, Y, Z, try to enlist the strength of X, Y, Z, then try to get out of the strength, try to get opportunities for those strength. I have told you about RNTCP. RNTCP has got 5 drugs. Can I do it with the 4 drug? Can I do it with the 3 drug? Can I do it with the 2 drug? Can I do it with 1 drug? Can I give drugless therapy? See, this much of opportunity is there. Suppose if I say RNTCP is for 6 months, you will get opportunity saying that can I do it for 5.5 months? Can I do it for 5 months? Can I do it for 4 months? Can I do it for 3 months? Can I do it for 2 months? Can I have a single dose medicine for tuberculosis? See, that much of opportunity you are getting. Suppose if I say, so when I talk about the opportunity, you have to think in terms of these 5 things. One is duration. One is combination of medicine. See, if there is 15 days duration, there is an opportunity for you to do it within 15 days. Suppose there are drugs is given for 3 times, there is an opportunity for you. Can you do it for 2 times? Can you do it for single day, single dosage? Is it possible? See, I will give you the example of vermicidal. Because formerly the vermicidal they used to give for 15 days. Then they came with some mebendazole. It will be given for 3 days, one BD for 3 days. Then they started, why I have to give it for 3 days? Then they came with the albendazole. It's a single dose medicine. Now you can say 200 mg of albendazole they are giving. Can I do it with 100 grams? Can I do it with 5 grams? Can I do it with 1 gram? Can I do it with the drugless? See, that much of that's why when you identify the problem, try to illustrate the problem in a proper way in the form of strength. After strength, you will get many opportunities. Try to enlist the opportunity and pick up one opportunity. For that opportunity, you give a beautiful strike on intervention, and that intervention becomes your intervention. That is the thing, important things. And for each thing, for each intervention, you have to give an justification. This is very, very important. So that's why I have put here the design. These are all the designs are there. Now, next slide. So I have told you methodology. So I have discussed the methodology. So when you talk about the methodology, I will summarize. First and foremost, to do a research, you have to work on data. Without data, you cannot do the research. So you have to get the data. So how to get the data? Without data, you cannot work and give the research answers. So here, when you try to collect the data, there are particular directions are given in research plan. There are particular direction. So you can collect the data in terms of, there are only 4 things are there. By doing research, you can collect the data. First. Second, you can collect the data by questioning the things, questioning about variables. You have seen in your house, the representative will come, sir, I wanted to give you a questionnaire. So we are manufacturing some soap, Vim or Rin or Rexona. So they will give you 5 or 6 questionnaires. And you have to take yes or no, whether you are using Rexona or not, yes or no, how many days you are using, since how many days you are using, whether you are happy with the order, whether you are happy with the presentation, whether you are happy with the. So they are collecting the data in the form of questionnaire. This is called questionnaire. First is experiment. Second one is questionnaire, by questionnaire. Third one, you can do it with survey also. Survey is one of the important things. The fourth one is you can do it with the interview. You cannot, for VIPs, you cannot give a questionnaire. For Prime Minister of India, you cannot give a questionnaire. For Prime Minister of London, you cannot give a questionnaire. What you have to give? You have to get an interview with him, and you have to collect the data by way of interview. So we are having experimentation, we are having interview, we are having questionnaire, and we are having observation or survey. Observation, we call it as observation methodology. So when I talk about methodology, we are having only 4 methodology. You cannot have 5, 6, 7, 8 methodologies. You are having only 4 methodologies. One, by experimenting. Second one, observation, experiment. Suppose if you wanted to do some new drug, you have to experiment. So that is experiment. Observation, clinical trial. All clinical trials are observation methodology. Clinical trials. Suppose if you wanted to know what is the prevalence of tuberculosis in a population, what you will do? You will go to house and house-wise survey you will do, and it is nothing but your questionnaire. Or otherwise, you wanted to know prevalence of diabetes in hypertensive patient. What you will do? You will go to the diabetes house and you take an interview of them, whether you are having diabetes for how many days, how many days, are you having blood pressure, all those things, data will be collected. So this is called collection of data. How you will collect the data? Please note, in a methodology, you have to write correctly what is the method you wanted to follow. There are only 4 methods: experimental method, observational method, and the interview method. The last but not the least is questionnaire method. So when I talk about questionnaire method, you have to formulate the questions. So questions, when you are formulating the questions, you cannot ask some direct questions. And when you are formulating the questions, please note these 5 things. Questions should be valid. Questions should be acceptable. This is very important. You cannot ask anything under the sky. It is not possible, because it will be a bad thing in research. You cannot ask. So, do you have an Audi car? Do you have a Benz car? Do you have a Maruti car? You cannot ask like that, because you cannot ask the personal things of the subject or patient. This is very important. You should ask only questions which is needed to collect the data about that problem. If it problem is disease, you have to ask only about the disease. Then the third one, whenever you collect the data, confidentiality. This is very important. Ethical behavior of the research. Confidentiality should be maintained. This is very important. Please note that. You cannot collect, you will collect the data and you will throw the data in a dustbin, or you will give the data to, because Farid has told that when you talk about data, primary data, secondary data, and all those things, okay, it is a secondary data, I will throw the data to the dustbin. You cannot do that. Am I right? So this is very important. So when you are collecting data, data should be valid, data should be acceptable, data confidentiality of the data should be maintained, and the last but not the least, data should be repeatable. Please note that. Suppose if I give a questionnaire to this patient and he will write all the answers, and after 24 hours, if somebody, Mr. X gives the same answer, same questionnaire, I should get same answers. Otherwise, it is called as bias. This is very important. Bias, personal bias. So please note that whenever you prepare a questionnaire, there should be, this is very important, repeatability in the questionnaire. So I will repeat. So acceptability is one of the parameter for questionnaire. Repeatability is one of the parameter for questionnaire. Then the validity is one of the parameter for the questionnaire, and acceptability is one of the parameters for the questionnaire. So this is method. So the data collection methodology. Where you are collecting the data, you have to specifically mention, because you are working not on the population, you are working on a sample. So specifically, you have to tell that I will collect the data from my college, or OPD of my college, IPD of my college, or sample. I will collect sample from special camp conducted for this purpose. Suppose if this one plan is clinical. So you have to specifically, precisely mention the area of your research, place of research. This is called sample collection area. This is very important. Number of samples. How many samples you are collecting? Number of data. How many data you want? You have discussed that about. I think Farid has discussed about. So sample, for to collect the sample, there is a formula. So this is Farid has discussed about that. Am I right? Yeah. So sample. So you have to go for incidence rate. You should know the prevalence rate, incident rate, prevalence rate, and the formula is there. Put the incident rate, prevalence rate there, and the standard error put 5% or 3% or 2%. You can put anything from 1% to 5%. You can put anything, and clearly come out that the number of sample you wanted correct. Suppose if it is for diabetes, the normal sample size is more than 300. Suppose if it is Saulia, it is around, if it is in Bangalore, it comes around 400. Suppose it is tuberculosis, and if it is in Mysore, it comes around about 45 something. Suppose it is Vata de Casa, it comes around 80 in Mysore, but in Bangalore, it may come around 120 or 130. So sample size is very important. Then, see, I have told how you are collecting, how you are taking sample, where exactly you are taking sample, and sample area, and number of samples. So you said I will only collect tuberculosis patient. Okay, now you have to clearly say there may be 100 number of diabetes patient, or there may be 100 number of tuberculosis patient. You cannot choose the patient as your wish. Oh, this is my neighbor, I will take him as a patient. He is my relation, I will take. You cannot. If you take it like that, it will be a bias sample. Bias sample. So you cannot take like this. You have to collect the patient by way of randomization. When I talk about randomization in research, randomization means you have to put all the patient in a framework or in a glass, name of the all patient in a glass, and you have to take the chit. Suppose 100 patients are there, you have to select the patient by way of lottery method. Lottery method. So lottery, you have to put all the names of the patient in a vessel, and you have to choose by lottery. And this is called randomization. To do randomization, we are having two techniques. One technique is called as lottery technique. The other technique is called as random book. Because in your PUC, you have heard about logarithm book. Like that, we are having a random book. In random book, they will write the numbers, and you have to choose a particular number. You have to assign number for the patient, and in the book, whatever the number comes, That number patient only you have to select. This is called randomization. Then sampling technique. Sampling technique is always you should not do the bias sampling. Bias sample is very, very bad in research. You have to do only randomized sampling. In randomized sampling, there are about 7 different methodologies are there. I will not talk about it because the time is very, very short for me. So you please go for simple random sampling, and it is called as SRS. Don't worry about other type of sampling. We are having quota sampling. We are having convenient sampling. We are having different sampling methods are there. We are not worried about it. But for your research, for simple research, simple random sampling is one of the best method which can be applied here. It is just you have to make sure that all patients in the area should come there and name them, number them. All the numbers should be put in a vessel and take the lottery. That's all. That's called a simple random sampling. Simple random sampling. The definition of simple random sampling is that sample, it is a beautiful sample wherein all the data are available, all the data are homogeneous, and all the data are having an equal opportunity to represent. All the data are having an equal opportunity to represent in the choice. What is the opportunity, the ratio? Ratio is, suppose the total number of data is n, the ratio is 1 by nth. 1 by nth means all are having equivocal representation. This is very, very important. All are having equivocal. So if you put all their names in the bottle or a vessel or a tub and take in a lottery, that means all will get equal opportunity to represent the sample, and selection is based on randomization. You cannot do the bias at all. This is very, very important. After that, so suppose in your thesis it is not possible, because you have to, in thesis or in your synopsis, you have to clearly define what type of patient you want. It's so it is called inclusive criteria. Inclusive criteria means, suppose if you wanted to work on diabetes, whether you wanted to take fresh case or chronic case. If it is fresh, you have to clearly say that, oh, I will take only fresh case. That means you have to delete other cases. Suppose if you wanted to take only fresh diabetes, you have to say age between 25 to 40, fresh. Suppose if you wanted to take only chronic cases, you have to write age between 40 to 60. If you wanted to take only the problematic cases, wherein Prameha Upadrava cases you wanted to take, then you have to say, I will only take chronic cases, because fresh cases you should avoid it, because Prameha Upadrava will not be there in fresh case. So you have to define the case. So defining the case on the basis of specificity is called as inclusive criteria. Specificity, because I need only this much of case. So specificity is based on 4 things. One, age-wise specificity. I will take only the patients who are between 20 to 40 or 40 to 60 or 60 to 80. See, age-wise specificity. The second one, diagnosis by specificity. This is very, very important. Inclusive criteria based on diagnosis. I will take the diabetic patient who are having FBS more than 160 to 200. So you have diagnosed it, and inclusive criteria based on the range of diagnosis. This is very, very beautiful. So when you say that specifically 200, within 200, I will take FBS within 200, FBS within 250, FBS within 300, you should take only cases of that only. You should delete all other cases. You should exclude all other cases. See, inclusive criteria. At least you put at least 2 to 3 points on inclusive criteria, and you have to write exclusive criteria also. This is methodology. Exclusive criteria. Which are the patients you wanted to delete? Most of our students are doing whatever you have written in inclusive criteria. Opposite to that is the exclusive criteria. It is not right. You have written that I will take only patients of 20 to 40. What is the exclusive criteria? More than 40, I will exclude. You should not write like that. You should not write like that. Please note that. I will repeat. Please note that whatever you have written in inclusive criteria, opposite to that will not be the exclusive criteria. So whatever, when I talk about inclusive criteria, the Samanya Siddhanta is inclusive criteria, Vishesha Siddhanta, Samanyam Ekatmakaram, Visheshasta Prataktat. The Vishesha Siddhanta, whichever you exclude is Vishesha Siddhanta. Vishesha Siddhanta is always exclusion. What you have to write? So you cannot write whichever is over 40 is excluded. No. The cases which are having systematic illnesses like nephropathy, I will exclude. Retinopathy, I will exclude. Or the chronic cases, I will exclude. You have to write like that. So at least you have to write the inclusive criteria 3 to 4, exclusive criteria 3 to 4. Then, so you have to discuss about in methodology, you have to discuss about data. In data, you have to write how you will collect the data, how many data you are collecting, how you are collecting the data, how many data you are collecting. Then, what is the methodology by which you are collecting the data you have to write. Then, where in which place you are writing, you are collecting the data. 4 things you have to write about data management. After that, you have to write about the data, specificity of the data, inclusive criteria, exclusive criteria, then diagnostic criteria. See, please note that this is about specificity of the data. When I talk about specificity of the data, diagnostic criteria, inclusive criteria, exclusive criteria. Diagnostic criteria in which you have to write the diagnostic tools. Suppose if it is radiology, you have to write, I will do the radiological investigation, X-ray or CT or MRI or SPECT or PET. You have to write all those things. You have to clearly say which part you have to taking X-ray. X-ray, just X-ray, PA view. Or chest X-ray, AP view. You have to write specifically. So radiological over blood investigations, biochemical parameters, you have to write. All the biochemical parameters which is related to the disease. I have seen many synopses where they will routinely write all hemogram, HPTC, DCESR and all other things. No, don't write like that. Please write which is specific to you. Because I have told it is diagnostic tool. Whichever assists you to diagnose the patient, you have to write only that thing. You cannot write all the things. Suppose if it is Taulia, you have to write about which examination? Can anybody talk? I think all of you are muted. So you have to talk about cholesterol. You have to talk about the cholesterol, content of the cholesterol, diagnostic criteria. So when I talk about specificity of data, inclusive criteria, exclusive criteria, diagnostic criteria. Okay. For the purpose of research, you need some tool, and these tools are called as assessment tools. Assessment tools. So how you will assess? How you will say that your treatment is successful? How you will analyze it? So you should have a assessment scales. For that, for psychological batteries are there, or you can develop your own scale. Because when I talk about qualitative analysis and quantitative analysis, for qualitative analysis, we don't have scale. For Ayurveda, for Brahma, we don't have scale. For Shiroshula, we don't have scale. For Samsa, Vesa, Veda, all Vedanas, we don't have scale. You have to develop your own scale. See, for Vedana, severe Vedana, it is 3. Medium Vedana, it is 2. Low Vedana, it is low Vedana, 0. Like that, you have to construct your own scale and standardize the scale. This is very important. Many of our researchers are doing scaling, but they are not standardizing. So you have to do a pilot study, and in the pilot study, you have to do the standardization of grading. So this is called assessment. So after assessment, you have to write something statistical analysis, because you have to prove your hypothesis. How you will prove? You need a statistical tool. So you have to write
in which statistical tool you are doing these things. Okay, the last but not the least part of methodology is ethical approval. So we will talk about ethical committee. Ethical committee is nothing but each and every college has got its own ethical committee. It is called institutional ethical committee, wherein the ethical committee will review or ethical committee will approve your clinical research study synopsis and your clinical study protocols. So you have to submit your synopsis to the ethical committee. They will review it and they will see whether these things are apt or not. Whatever you have written is apt. Whatever you have written is beneficial to the patient, because the ethical committee has got a right to discuss beneficiency versus maleficiency. If it is maleficent, if it is not beneficial to the patient, they will say you don't do it. So this is very, very important. There is a scientific committee in your college, and scientific committee will oversee the scientific writing, scientific skill of your synopsis, but ethical committee will oversee whether the project is beneficial to the patient, the project is beneficial to the science, the project is beneficial to the society. This is very, very important for you. So you have to do the patient, you have to do the project on the basis of beneficiency to the patient, beneficiency to the science, beneficiency to the sample, or beneficiency to the population. Whether it is beneficial or not, it is reviewed by the ethical committee. So ethical committee, this is composition of ethical committee. We will not talk about that. This presentation, I will hand it over to the Atrimed people, and they will send all these presentation to you. You can see whenever you wanted to see this presentation, you can review it and you can learn about the writing the research. So responsibility of the ethical committee will oversee the ethical behavior of the research, whether the research synopsis is written in ethical way, and whether the protocols are standard, and the outcome of the protocol or outcome of the synopsis is beneficial to the society, beneficial to the patient. This is only the duty of the committee. So they will see, because these are the very, very important when you are writing the synopsis. This is very important. You have to, first and foremost step for the ethical committee is that first you have to submit it properly in the protocol to the committee. Then when you are submitting, there should be informed consent form. Please note that without consent you cannot do a clinical trial at all, because the subjects are not animals, subjects are not slaves, so they have to give the consent. Consent is very, very important, because it is legally bounded. Suppose something happened in the research, you will be safe if you are taken a consent form. Please note that you have to write a consent form. Then recruitment procedures, data collection. Recruitment means data collection procedures. Whether you have done it randomizedly, whether you have taken sufficient number of samples, whether the samples collected are perfect or not. These things they will review it and they will see whether they have taken written information, written consent from the patient. Then the safety of the information, safety and efficacy of the problem. Then suppose if it is safe also, whether there is any monetary benefit to the researcher, because they will see, because there should not be any adverse reaction, there should not be any monetary things. You cannot take some money from the patient. This is very, very important. So they will ask about the safety of the safety of the intervention. Then talk about the adverse reactions, then drug interactions, then drug reactions. All those things they will ask you. This is very important. Then CV of the investigator, whether the researcher is qualified or not. This is very, very important. Whether the researcher is having a PI, whether funding agency is G9 or not. All those things can be reviewed by the committee. Then approval. So after reviewing, they will give you the approval, and approval should be signed by the chairperson and all the members. Please note that there should be signed copy of the chairman, and it should be given to you. If you are not getting that form, you should not do the research at all. Okay, this is very important. Suppose there should be always the odd number of members. Why odd numbers? 9, 7, 11, like that members should be there, because suppose if there is a difference between the members, it should be put on the votes, and whoever get the majority, that decision will be taken. The decision will be taken on the majority. This is very important. Then animal, so suppose you are doing in vivo, in vitro, or animal studies or cell line studies, you should take permission from a animal ethical committee. This is very important. This is about freedom of animal welfare, ethical committee, all those things we have talked. So you can read it out because time is not there. So I will discuss about statistics now, because this is very important. Statistics is very important. Statistics is very important for you. So, so this is about statistics. We will not talk about that. So here you have to write this, because when I talk about statistics, you have to write these things, because you have to write statistical tools in your this one. So statistical tools, you have to write first about descriptive statistics. Whether you are doing some graphs, whether you are doing some tables, whether you are collecting the data in the form of table, or whether you are collecting the data and you are representing data in the form of graphs, you said say descriptive statistics will be utilized. When table is there, when the graphs are there, when you are using SPSS software, there is a software is there. If you put the data in the software, SPSS will give you a graph. SPSS, there is notepad is there, graph pad is there, SPSS is there. So all those will give you the graphs. So when you are utilizing this, say please write the first heading that descriptive statistics will be used for the study. That means when you write descriptive statistics, you will say data analysis of the data in the form of tables, in the form of graphs. There are many graphs are there. Pie diagram is there, bar diagram is there, horizontal bar diagram is there, vertical bar diagram is there, parallelogram is there, histogram is there. All those includes under descriptive statistics. So you please write, I will utilize the descriptive statistics. That means you are taking bar graph, you are taking pie graph, you are taking tables, different tables, horizontal bar diagram, vertical bar diagram, pie diagram, and all those things you are talking. Then second, you have to write about inferential data. What does it mean? So you have given your protocol to the IEC committee. IEC committee has given the approval. You have started the research. You have started the research without any bias, and you got one of the best results. But my question is that whether the result you obtain may be by chance or by choice. You got my point? It may be by chance. Can you give me a guarantee that if I give Dawais Loha for 10 Pandu patients, can you give me guarantee that 100% you will get success? There is a probability that if you don't give the Dawais Loha, also the patient may be okay. That is called chance occurrence and choice occurrence. So in research, we need choice occurrence, not chance occurrence. We have to say, when I talk about the inferential statistics, we are calculating the results you obtain is by chance or by chance. So for that, we are having many tests. We are having T test, we are having F test, we are having Z test, we are having many tests are there. Those tests we are discussing here. Those tests we are discussing here. So usually for quantitative data, quantitative means the data collected in the form of a number. For quantitative data, we will use T test, we will use Z test, we will use the paired t-test, unpaired t-test, z-test, and normal test, all those things for quantitative data. Suppose the number of data is 30. Number of data is 30. So you cannot use the z-test because you should use a small test. Small test means you can say t-test. Suppose the number of data is more than 30, you cannot use small test. You have to use big test. Big test is z-test. After T, Z comes. That's why it is called big test. It is called normal test, big test, or Z test. So if data, so depending upon the number of data, you have to choose your test. If there are two groups, you can say paired test, T test, paired T test. If there is only one group, unpaired, because you cannot pair it. So unpaired T test. Suppose of the ranks, in the form of class, in the form of grades, and it is called qualitative. Qualitative. Those are for higher groups. 25 groups are there, not for you. But for your convenience, for Atrimed researchers, I think qualitative, quantitative, you will have only two groups. Usually, most of the time, 99%, if it is quantitative, you have to write T test, Z test, F test. If it is qualitative, you have to write chi-square test. Please note, chi-square test, Wilcoxon ranks test, Wilcoxon sign test, U test. Within these 8 tests, you can finish it off your statistical analysis. Suppose you are having assessment interval 3 to 4 times, 15 days, 30 days, 45 days, 60 days, follow up after 120 days. See, follow up also very, very important. Follow up after. So you are doing assessment of the same patient at different intervals at different times. Then you cannot use this one. Then you should go for repeated ANOVA. Repeated ANOVA. These are all the 2 minutes. 2 minutes. So these are about the statistical analysis for your patient, for your this one. Last thing, hypothesis testing. The testing of the hypothesis we are doing analysis of the T test, Z test, F test, and it is called hypothesis testing. This is very, very important. So these are the things. So this is one of. So I will show you one of the this one synopsis. So synopsis, you should have an introduction. I will finish it up within 3 to 5 minutes. Is it okay? I just wanted to show you a sample synopsis. Sample synopsis. With the sample synopsis, I wanted to conclude. See, first you have to write the introduction. In introduction, you have to write, before introduction, you have to write a title. For title, there should be a problem, there should be a design, there should be intervention. After that, you have to write the introduction. After introduction, you have to write the need of the study. In the introduction, you cannot write Ayurveda is holistic, Ayurveda is 5,000 years old, Ayurveda is developed by Brahma, and all those things, all things you should not write. First and foremost thing is that define problem. Definition of the problem. So you have to start your introduction with the definition of problem, then prevalence of the problem, incidence of the problem, prevalence of the problem, justification of the problem, then status of the problem. Status of the problem is what is the status now? Whether for that problem there is a treatment available, and if treatment is available, what type of treatment? What is the diagnostic criteria? All this is called status of the problem. and SWOT analysis of the problem. Then you have to introduce your intervention. Then intervention, you have to talk about the drug, and after the drug, you have to clearly say the probable outcome of the problem. All these things you have to write within the 200 words. That's all. 200 to 250 words. And Atrimed has given a beautiful protocol for you. According to that, you have to write the introduction, did of the study, and after that you have to write a literary review. See, previous works and all those things includes under literary review. See it here. Then you have to write the methodology. You have to write the objective of the study, aims and objective of the study. Then materials and methods is nothing but methodology, source of data, a source of drug. Then inclusive criteria, exclusive criteria. See, all those things are there. Inclusive criteria, exclusive criteria. See, exclusive criteria, diagnostic criteria, nothing but laboratory investigation, and assessment criteria. See here, assessment criteria. Then last but not the least, about the references. You have to write the references in any one of the format. Any one of the formats. There are formats are there. Formats are available. You can write. I don't know. You have to whether you have to write in some format. You have to write. Please write in some of the format. So if you don't know the format, I will send the format PPTs to Atrimed people, and Atrimed people will send all those things to you. Don't worry about it. So sorry for inconvenience. So please pardon me because I had my boss today visited my office. That's why I came late. Sorry, I think I have done justice to you today. So special thanks to Rishikesh Namle, my good old friend and one of the greatest entrepreneur in Ayurveda. And my special thanks to Chandrakala Madam, Lata Madam, and Prithvi for giving me an opportunity. So thank you, one and all, Prithvi, Damle, and all Atrimed people. And my special thanks to Fareen also because she has taken the lead today by starting this presentation. So thank you, one and all. Before I exit, best of luck to you. Hats off to you. Please, 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 please write synopsis today and send the synopsis within the stipulated time, and please give an overwhelming support for this beautiful beautiful activity of Atrimed. I want such many activities which are educationally oriented, which are innovative in future days to come. Thank you, Prithvi. I hand over it to Prithvi.
Thank you so much, Dr. Shinoy. So now we will be taking a short lunch break. All of you can join us back at after 2:15 to listen to Dr. Pratibha Nadig talk about the overview of protocol writing. She will be covering various aspects of protocol writing, such as the planning of a protocol, structuring, and how to write a clinical protocol. Thank you all for your participation, and we will be ending the meeting now. You can join us back at 2:20 p.m. Thank you, everybody, for your participation.
ಸರಿ ಬಂತ?
ಹ?
ಸರಿ ಬಂತ? ನಾನು ಪ್ರಿಪೇರ್ ಆಗಿಲ್ಲ.